All right. Okay, so I am going to give this lecture through Google Air Hangout, so hopefully this will work for everybody. I unfortunately can't do full screen for the lecture slides, otherwise they don't actually work out. So I'm going to have to do it through this way. Hopefully you'll be able to see everything without any problems. Okay. All right, let me just get started here. Okay, so today's lecture is on biological molecules. So this is chapter 2 of your textbook. A lot of this, again, should be review from, in part, high school, and in part, some of the stuff that you covered in cell biology last year. So really, what we're going to be going through is a lot of the chemistry behind what's happening in a cell, because without understanding the chemistry behind it, you really can't understand how different components of the cell are going to interact with one another. We'll highlight the difference between hydrophobic and hydrophilic molecules, how water plays into all of this, the explanation behind acids and bases. Again, this should absolutely be review, but this is quite important in a biological system. The last half of the lecture is going to be focusing on really the 4 major groups of biological macromolecules that we see in the cell, how these macromolecules are put together through the process of polymerization, and generally what their functions are in the cell. So really, to understand how a cell works, you have to really start to break it down into really tiny pieces to understand all the complex interactions that are taking place. So at the heart, you have atoms and you have molecules which come together to form more complex structures, which can polymerize to create even more complex structures, which can then sort of fold or interact with one another. And ultimately, you get a lot of the really complex functioning that you see in a cell. So understanding the chemistry of how these things actually come together is really critical to understand how a cell is going to function. So this should be 100% review. The very smallest component in a cell is going to be your atom. So these would be all those elements that you would see in the periodic table. This is the basic unit of matter. So you're going to have a central nucleus with your protons and your neutrons, and then surrounding that nucleus, you're going to have a cloud of negatively charged electrons. And those electrons are going to interact with the outer electron layers of other atoms to form molecules. So these are stable combinations of atoms that are typically held together by covalent bonds. They can be the same atom or they can be different. If they're different, we often refer to that as a compound. And compounds can also have different atoms held together by more than just covalent bonds, so by other types of bonds that we'll talk about. So central to human biology is the covalent bond. That's really what's keeping, it's the strongest bond that we've got in our cells. So if you have here in blue the nucleus of a cell, then the outer layer, the outer valence layer of the electrons, you have, in this case, 7 electrons, so that would be equivalent to either a fluorine atom or a chlorine atom. Some review, the outer valence of an atom is going to be happy if it has 2 electrons or 8 electrons. So it's always going to be trying to seek out partners that have unpaired electrons to try and complete that outer layer, typically to get it up to 8, in some cases to get it to 2. So in the case of chlorine, for example, you have 7 outer electrons, so it's looking for its 8th. If it were to meet another chlorine atom, as you see over here, those unpaired 7th electrons can actually come together and pair off, and now you have equal sharing of these 2 electrons to complete the outer electron valence of both of these molecules, and that makes a very stable bond. So in cases where you actually have the same molecule the same atom coming together, so in this case the 2 chlorine atoms that are sharing that pair of electrons, you have what's referred to as a non-polar covalent bond, meaning each of those nuclei are equally pulling those electrons. So it's a very fair sharing of the electrons across the two atoms. But in any case where you have two different atoms coming together, where one is going to have more protons in its nucleus than the other, you're going to have sort of an uneven sharing of those electrons. So even though they are being shared, and you're having sort of completion of the outer valence level, one of those molecules is going to be slightly more electronegative than the other. So in example B here, you have the chlorine atom, which is going to be a more electronegative atom than the hydrogen, so it's pulling the electrons closer to itself. So even though this is a covalent bond, it's what we call a polar covalent bond. So you have a slightly negative end to this molecule and a slightly positive end to this molecule. But this is still a very stable covalent bond. Covalent bonds require quite a bit of energy to be able to be broken apart, and that's why they're so useful in a biological system. In probably in the most unequal of sharing electrons, you have the ionic bond. So in this case, again, two different atoms are coming together to fulfill their outer electron valence. So chlorine in this case is looking to get to 8, sodium is looking to get to 2. But in this case, the draw is so big, the electric force coming from the chlorine nucleus is so strong that it's actually poaching that electron from the sodium atom. So you have a complete transfer of one of the electrons into the outer valence of chlorine. And now what you end up getting is actually two ions. You have the sodium ion and the chloride ion, who obviously are still attracted to each other in the form of an ionic bond. Polar molecules. So any molecule that has a covalent bond, if I just go back, to this previous one over here, this would be a polar molecule where you have a charge on one end and a different charge on the other. So it's an asymmetric distribution of electric charge. And these are the molecules that are reactive in a cell. So these are the ones that are going to take part in hydrogen bonding, sort of interactions with other molecules. So water is the most common polar molecule that we find in our body, and it can involve itself in a lot of different chemical reactions with proteins, with amino acids, with sugars, you name it. Essentially, any molecule that has an oxygen, a nitrogen, or a sulfur group is going to be a polar molecule because these are highly electronegative atoms that are going to set up this sort of polar covalent bond in any molecule they take part in. Conversely, we also have quite a few molecules in our body that are nonpolar, so they lack polarized bonds. So they do not have this electronegative atom setting up this dipolar situation. And technically, these are really sort of inert molecules in our body. So these are the fats, the sort of waxes, for example, carbon dioxide even. They're not going to react typically with anything else in the body. And they're for the most part made up of carbons and hydrogens. So it's important to note, though, that a lot of the biological molecules are going to have different components of themselves that have polar activities as well as nonpolar activities. And that's going to help with different folding and different interactions between these different molecules. So I already spoke a couple slides ago about ions in case of ionic bonding, where you have a poaching of an electron here, but you maintain this electrostatic interaction between these two atoms. That atom which is taking on sort of the positive charge is what we refer to as the cation, and that which is taking on the negative charge is the anion. But it's important to note that these are still very stable. Even though they've got a charge, they're very stable, and that's because their outer electron shell has reached a stable state. So in this case, 8, in this case, 0, or 2, if you count the 2 that are on with chlorine. Free radicals, on the other hand, are unstable atoms that have an unpaired electron. So they have either lost or gained an electron in such a fashion that they do not have a stable outer valence level. And this makes them highly reactive, so they are constantly looking for somebody to either donate an electron to or to yank an electron off of. These are highly prevalent in disease states, which you're going to see in a couple of slides, but they are made throughout our body on a regular basis. So through the process of metabolism, we are making free radicals at all times, but we do have a series of antioxidant systems in our body which are capable of trying to remove free radicals that are being formed. If we cannot clear these free radicals, they can be quite damaging. So they can damage macromolecules such as DNA, for example, and cause mutations. And there's quite a bit of suggestion that free radical formation plays an important role in the process of aging. So as I mentioned, free radicals are really recognized as one of the major contributors to disease, and that's why there's a big booming market, at least in the nutraceutical world, in the formation of antioxidants to try and combat free radical formation. So as I said, free radicals can happen in our body naturally. So through the process of inflammation, we can get the generation of free radicals. Through the process of respiration in the mitochondria, we generate free radicals. And typically our body is capable of handling normal levels of free radical formation with antioxidant systems that we have. We run into problems, though, when we start seeing sort of environmental exposures which increase the amount of free radicals. So UV light can cause the formation of free radicals, ionizing radiation, smoking, and pollution are all known to cause free radicals. And then you start to sort of have an accumulation of these damaging molecules, which can cause DNA damage. A whole slew of different diseases which have been linked to the formation of free radicals. I'm just going to play this video here for you. Get it to work.
Tissue cells. Entering a tissue cell through the cell membrane. There are many elements in our body that comprise molecules. An electron from the outer bonding shell of one element forms a bond with the outer bonding second element, forming what is called a chemical bond. During oxidation, an electron can be knocked out of the chemical bond, resulting in a highly reactive free radical, where an unshared electron occurs. Oxidation factors include poor diet, pollution, drugs, radiation, stress, injury, aging, and infection. Disease risk include cancer, heart disease, arthritis, autoimmune disease, and over 90 other diseases. This free radical can capture electrons from normal healthy molecules and create free radicals that damage the healthy molecules, such as molecules found in DNA. The damaged cell can release free radicals and continue the effects of oxidative stress to surrounding cells. To prevent or reverse these very destructive processes, the body constantly needs a reservoir of antioxidant molecules. Antioxidants are important in defending against free radicals. Antioxidant molecules have extra electrons and can supply electrons to neutralize free radicals. The antioxidants travel through blood vessels to reach the damaged cells. The antioxidant molecule will supply the free radical with an electron to repair and stabilize the chemical bond. In supplying electrons, the antioxidant molecules do not themselves become free radicals. Frequent healthy dosages of a variety of antioxidants will assure this destructive oxidation process will be minimized, reducing the potential for cancer, heart disease, autoimmune disease, and diabetes, not to mention their importance in anti-aging.
So other than covalent bonds, probably the other most important type of bond that we're going to see in biological systems is the hydrogen bond. So a hydrogen bond is not a sturdy bond, so it's not actually sharing any electrons. It is an attraction of electrons from two polar or two. On this side, you have a molecule in which the carbon is bound to an oxygen. The oxygen is a more electronegative atom, so it's pulling the electrons towards itself. So it sets up this slightly negative charge. Over here, you have a nitrogen bound to a hydrogen, covalently bound. Then the nitrogen is the electronegative atom, pulling the hydrogen's electrons towards itself, creating a slightly positive charge on hydrogen. And now this slightly positively charged hydrogen is going to interact with a slightly negative oxygen here. So this is really sort of an electrostatic attraction. There's no actual sharing of electrons here. It is not as strong of a bond as a covalent bond, so they can actually be broken easier, which is why you're going to find them in some really important places. So for example, in water, all the different molecules of water are interacting with each other through hydrogen bonds. Another really important place that you're going to see hydrogen bonds is between the bases of the DNA, two strands of DNA. And you want sort of this weaker type of a bond. You got covalent bonds here, for example. You'd have to put an enormous amount of energy in to be able to pull apart those two strands of DNA to replicate the DNA. So every time a cell had to replicate itself. By using hydrogen bonds to keep these two strands together, a very small amount of energy, we can pull those two pieces apart. So these concepts should be certainly review for you, the concept of hydrophilic versus hydrophobic. So hydrophilic is really referring to anything that is water-loving. Really what you're talking about here are molecules that can take part in hydrogen bonding. So these are going to be molecules that have some polar covalent bonds inside of them. When you have these ability to take part in hydrogen bonding, it can hydrogen bond with surrounding water molecules, and it can allow those molecules to become soluble in water. So when you think about sugar, for example, when you pour sugar into a cup of water, you can stir it and it will dissolve, and that's because the sugar is actually capable of taking part in hydrogen bonding with its surrounding water molecules. So you see hydrophilic molecules are really, as I said, sort of polar molecules. As far as biological types of molecules that we're talking about, sugars that I just described, several amino acids have hydrophilic side chains, which allow them to also be soluble in water. Conversely, you can have hydrophobic molecules, and these are sort of water-fearing molecules, so they do not have polar, so they're very neutral in charge, and as a result, they cannot take part in hydrogen bonding. And really what you end up seeing is they become insoluble in water. So if you think about fat droplets on water, the fat is hydrophobic, it can't actually take part in hydrogen bonding, so it can't actually become soluble, and instead it sort of clumps together. So fats, steroids, we're going to talk about a few of those different things a little bit later on in this lecture. One type of really interesting hydrophobic interaction that you will see that does actually have some really important biological significance is what's called van der Waals interactions. So if you have two hydrophobic molecules, or two portions of a hydrophobic molecule that come in very close contact with one another, even though they don't have an established charge that stays put all the time, electrons, as you know, are moving constantly around the nucleus. So at any given time, you could have an atom that has all of the electrons on one side of the atom and no electrons on the other. Sort of get transient dipole set up across the atom. And this can happen in hydrophobic molecules. You'll have these transient moments in which poles are set up, and you get this electrostatic interaction between these two molecules. And what makes this actually become more of a stable interactive system is it can actually sort of like dominoes set off the creation of dipole atoms in all the neighboring atoms, all the neighboring hydrophobic atoms that are side by side. And so you get this very weak. It does not take much energy at all to break this apart. But they do have a biological relevance. So for example, when antibodies interact with antigens on, for example, on viral antigens, in some cases you have two hydrophobic surfaces coming together and they actually are interacting using van der Waals interactions. So as I've alluded to, water is really what's making up the bulk of our body. And as a result, the chemistry of water and how water interacts with different biological molecules is highly important. So you have two covalent bonds in the molecule of water. So you have oxygen creating two covalent bonds with two hydrogen molecules. Because oxygen is highly electronegative, it is pulling those electrons towards itself, creating two positively charged hydrogen atoms, which can take part in hydrogen bonds. Is also negatively charged, so it can also take part in hydrogen bonding as well. The covalent bonds that are holding the individual molecules of water together are very stable, so it actually takes quite a bit of heat to evaporate. So you can think about that logistically at your house when you're trying to boil some water. Because of its capabilities in doing all of this hydrogen bonding, it is a wonderful solvent. Substances that are capable of becoming soluble inside of it. Acids and bases and buffers. Again, we're not talking about these over the course. That biological systems are highly attuned to work at very specific pHs. And when we refer to pH, what we're referring to is free hydrogen protons that are available. So acids are compounds that are capable of releasing a proton in the form of a hydrogen without an electron. Bases are capable of accepting those protons, and amphoteric molecules have the ability to act as either a base by accepting a proton or as an acid and donating a proton. So water is an example of that. In both cases, it's a fairly weak acid and base, but under the right circumstances, it is capable of donating a proton and becoming a base, or it's capable of accepting a proton and becoming an acid. So as I said, biological processes are very sensitive to pH because you can imagine that these free-floating protons would drastically change the chemistry, all of those types of chemical reactions that I've just been talking about. And so in order to make sure that pH stays relatively stable throughout the human body, we have a variety of buffer systems that are built in, which their entire purpose is really to try and maintain a very constant pH so that biological systems can be running smoothly. So one of the best examples of a buffer system is the bicarbonate carbonic acid buffering system in blood. So you have a bicarbonate ion, which is capable of mopping up any free protons that might be let loose into the blood system to become acid, or going the other way if need be. Okay, so this should not be news to anybody, but carbon is really central to all organic compounds. I'm not sure if you guys need to take organic chemistry, but really the study of any compound that has carbon in it. So what makes carbon wonderful is that it's capable of bonding to up to four different atoms. So it has an outer valence electron count of 4. It is a very stable atom. It is capable of making a really strong backbone for a lot of different structures, and it's highly, highly stable. So very simple carbon molecules are called hydrocarbons, and these are molecules that strictly have carbon and hydrogen making them up. So you can see them over here. These are the hydrocarbons. We typically do not see these in biological or living systems. Instead, these really make up the bulk of our fossil fuels that we think about when we are trying to drive our cars or heat our houses, for example. Instead, in biological systems, if you look again at some of these chains, what you have is that some of these hydrogens are actually replaced by functional groups. So those functional groups could be any of those that are seen here. And these functional groups, as you can see, for the most part, have polar atoms involved in them. So they're allowing these molecules to actually interact with one another in one way or another. All right, so let's talk more broadly about the types of molecules that we would see in a living system. So first and foremost, you have your series of macromolecules. These are the big molecules that are making up our body. And when we're talking about these, we're talking about carbohydrates, lipids, nucleic acids, and proteins. So really, this is what our body is made of. To get these macromolecules, these are essentially polymers of smaller building blocks. So in the case of carbohydrates, they're made up of building blocks of sugars. In macromolecule lipids, made up of fatty acids. Nucleic acids are made up of nucleotides, and proteins are made up of amino acids. So in a cell, not only would you have your macromolecules, but you would have all the building blocks to make those macromolecules. Over the course, even next lecture, actually, on Wednesday, you guys are going to be talking a lot about metabolic pathways happening in the cell, which are ultimately aimed at either making energy, breaking down sugars to make energy, or conversely using that energy to build different macromolecules, for example, in the cell. So those are metabolic pathways that you'll be talking about. And along those metabolic pathways, some products will be formed that will be sort of transient in nature. Some of them do not. Some of them also are a specific class of molecules that we'll see in the cell. And then finally, we have a small set of sort of miscellaneous molecules. Are all very important for function of a cell, but don't neatly fit into the box of the three categories above. So the one we talk a lot about in this course is ATP. So this is how we essentially are shuttling energy around the cell. But it also includes things like vitamins or cofactors that might be needed in order for some of the metabolic pathways to take place. So let's talk about the first class of macromolecules. So As I said, these are large structural molecules that have function. And there's the four categories that you can see here. So the first of them are carbohydrates. So carbohydrates are polymers made up of simple sugars. And really, carbohydrates are where we are getting our energy from. So we typically eat carbohydrates to get energy because we break them down to sugar, to the simplest of sugars, and use that energy to carry out a variety of purposes in our body. But we can use some carbohydrates, more plants than humans, but certainly structural components can be made out of carbohydrates as well. Lipids are a class of non-polar molecules within our body. They can act as an energy storage site. So if we're eating too many carbs and we are taking in too much sugar, instead of using all of that, we can actually store it in the piece of fats. But they also play very important structural roles. So the phospholipid bilayer of the membrane, the cell membrane is made up of lipids, and some hormones are actually made up of lipids as well. Nucleic acids are long polymers of nucleotides that essentially their purpose is to store and transmit genetic information. So it's storing our genetic information in the context of DNA, and it's actually translating that information into the formation of proteins through RNA. And finally, a really big class of macromolecules is proteins. And proteins are polymers of amino acids, and they have enormous numbers of functions, which we're really talking about. So let's start off by talking about the carbohydrates. So carbohydrates, as I said, this is really where we are getting our energy from, and can, in some cases, be durable building materials. So carbohydrates are polymers of sugars. So what you're looking at when you talk about a sugar is a molecule that is made up of a series of carbons down to a hydroxyl group and down to a hydrogen in a chain. And this is going to repeat itself over and over, anywhere from 2 to 7 molecules in length. And there's two primary structures that you're going to see. So somewhere embedded within the sugar molecule, you're going to have a carbonyl group, which is a carbon double bonded to an oxygen. If that carbonyl group is found within the actual sugar, as you can see here, that makes a ketone, and we call this a ketose sugar. And the best example of that is fructose. On the other hand, if the carbonyl group is found at the very end of the sugar molecule, like you see here, making aldehyde, this is what we refer to as an aldose sugar, and the best example of that is glucose. So certainly for simplistic sake, we often will depict sugars in a linear fashion like this. But typically in a biological setting, they will spontaneously form these ring-like structures here. So usually when you're talking about glucose and fructose, you're talking about a ring-like structure. That being said, you absolutely can get linear sugars in a biological system, and particularly in the case of diabetes, for example, we see quite a bit of the linear sugars, which can actually cause a lot of problems in functioning of a lot of really normal proteins, so for example, human protein. So those simple sugars, so this would be an individual sugar here and here, they are going to often link up with one another to become, in some cases, disaccharides. So that would be a combination of two sugars bound together. Oligosaccharides, which are anywhere from 2 to 10 sugars bound together, and polysaccharides, which can be hundreds of sugars in them. And when these sugars come together, the type of bond that they take part in is what's called a glycosidic bond. So you can see that here between the two carbons, you have an oxygen. So obviously the most simple sugar is going to be either glucose, fructose, or galactose, for example. But you can get combinations of these sugars coming together in pairs called disaccharides. So sucrose is one that we all know quite a bit about. It's typically what we're getting at table sugar, which is made up of a glucose and a fructose. Lactose, which you've also heard about. So this is a glucose down to a galactose, and there's a variety of different combinations that you can make up. We just said oligosaccharides are going to be chains similar to this that are anywhere from 2 to 10 in length, and they often will play a role not so much in energy providing, but instead in acting as sort of surface markers on cells bound to lipids or cell representation. Saccharides, this is typically how we actually ingest a lot of our sugars. These are more complex sugars to be taken in, or more complex carbohydrates to be taken in. So you can get glycogen, which is an animal product of branched glucose polymers. So each one of those sugars is a glucose. And this is a really good animal storehouse for surplus energy. A plant sort of equivalent is starch. So you get potatoes, for example. This is how the glucose is coming in from potatoes. And the third type of sort of polysaccharide that we will see throughout this course is what we refer to as GAGs for short. So these are we are structural polysaccharides. So they are not really polysaccharides that we're using as an energy source. But they're complex, they're highly branched, and we will typically see these on the cell surface or in the extracellular space. And in a couple of lectures we'll talk at length about this. Okay. For interest's sake, I just threw in a couple of slides here just in context of what a good carb versus a bad carb is. You hear that a lot in the news. You hear that a lot in sort of everyday speak. But in relation to molecular biology, what is it that we're actually talking about? So when we talk about good carbs, what we're talking about is actually complex carbs. So those highly branched carbohydrates that are typically coming in with other nutrients. So not strictly sugars, but are coming in with other vitamins and minerals as we're eating them. And so that's what we refer to as the complex carbs. Bad carbs are simple carbs. So these are those monosaccharides and disaccharides, very simple, very easy to break down, and usually coming into the body with very few other nutrients. And so how and why are these so good or so bad? This comes down to the concept of the glycemic index. So typically when we take in a complex carbohydrate, so we have a big long string of sugar molecules that are bound to one another, in order for our body to be able to capture energy that is stored within those sugars, we have a bunch of different bonds here that we need to break down. And so what happens is that over time, our body has to work for some time to do that, to break down that complex sugar. And so we never get this really high spike in blood sugar levels. We get sort of a low, constant release of sugar and energy in our bodies. Conversely, when we take in very simple monosaccharides and disaccharides, so we're drinking pop and eating chocolate bars, we're taking in a lot of these, our body can very quickly break down these molecules, so we get these spikes in glucose. We won't actually need that much energy at that time, so a lot of that is actually going to be converted and stored as fat. And so that's what we refer to as the bad carbs. Okay, the next class of macromolecules is lipids. So these are a diverse group of nonpolar molecules, and really the three primary classes are fats, steroids, and phospholipids. So if we look at the structure of a fat molecule, really the purpose of these is to store energy long term. So remember we talked about taking in that pop or the chocolate bar, you get this spike of sugar that our body tries to break down and make energy with, and then we realize we don't actually need that energy, so instead we store that energy in the form of fats. And so this is what they actually look like. So you have a glycerol moiety over here, and attached to that glycerol moiety you have 3 fatty acid tails. And here's just a zoomed in of what that fatty acid tail looks like. So you have a series of carbons bound to hydrogens, and this is going to be the hydrophobic area. And you do have a hydrophilic area, just one little piece right here, where you have a hydroxyl group and a carbonyl group here, and that's what's going to bind to the glycerol moiety. These fatty acids, so they can be what's referred to as saturated, which is what you're actually seeing here. So that means that every carbon is fully bound to as many hydrogens as possible. Or you can actually have unsaturated, where you have some double bonds taking place between the carbons, so they're not fully saturated by hydrogens. Another form of lipids are steroids, so these are hormones that we're making. And so cholesterol, which can then be further developed into testosterone or estrogen. These lipids are really interesting. These lipids are 4-ring hydrocarbon skeletons, and again, these are non-polar. And then the type of lipids you guys have probably heard quite a bit about in cell biology are the phospholipids. So they actually look very similar to the fats that you were just seeing back here. So remember you had the glycerol moiety with the 3 fatty acid tails. Now if we look at the phospholipid, here's that glycerol molecule right here. And instead of 3, you've got 2 fatty acid chains attached to it. Instead of a 3rd fatty acid chain, you have a phosphate group, which is bound to some small polar group. And this is what you're actually going to be seeing in the phospholipid bilayer of a cell membrane. And the reason that you want to have this sort of polar group both over here, and again, remember, in a phospholipid bilayer, you're going to have another one of these sort of mirror imaged over here, and another polar group over here. And that's because you're going to have water on either side of the cell membrane. So this allows the cell membrane to exist in a living system. The 3rd class of macromolecules are nucleic acids. So nucleic acids are polymers of nucleotides, which I'll talk about in a second. And there's really 2 forms of nucleic acid. There's DNA, which is what we see in the nucleus, and that's storing our genetic information, and RNA, which is how we are transmitting that genetic information into formation of proteins. So, as I said, these are usually long strings of nucleotides, and this is what the standard structure of a nucleotide looks like right here. So at the core, you have a ribose sugar. On one side of that ribose sugar, you're going to have a phosphate group, and on the other side, you're going to have a nitrogenous base. And there are 5 different bases that exist in human cells, 2 of them being purines and 3 of them being pyrimidines. And this is what makes up your G, your A, your C, and your T in the case of DNA, or U in the case of RNA, looking at sort of a string of genetic information. And you can see that each of these bases are slightly different. So when they are coming together, so if we think about the double strand of DNA, so if you just look actually up here in the corner of my slides, this is your double helix of DNA. So you have a backbone here in red, and then you have the bases projecting, making sort of the rungs of the ladder. And this is what this really looks like. You have your sugars attached to your phosphate. That phosphate group is going to bind to the sugar of another nucleotide. There's its phosphate group, bound to the sugar of another nucleotide. There's its phosphate group, etc. And this is what we refer to as a sugar-phosphate backbone. And then you see these bases sort of projecting out to the side. And they are going to make hydrogen bonds with neighboring bases on a second strand of DNA over here. There are other nucleotides that exist, and some of them have completely different functions. So ATP is one of those sort of other molecules that we talked about, but it is actually made up of a nucleotide. So here again, you can see sort of the ribose sugar in the middle. You have a nitrogenous base over here, adenine, and then you have 3 phosphate groups. So that's the A triphosphate that you're seeing here. And we'll be talking quite a bit about that starting next lecture. And the last class of macromolecules, one that is highly important in this course, is proteins. And this is because these are the macromolecules that are performing virtually all of the cell functions or cell activities. So all of those molecular tools and machines that we saw in the video last week essentially are proteins doing their job. So the major functions of proteins include acting as enzymes, so catalyzing reactions in a cell. They transport things around the cell and around the body. Some hormones are proteins. A lot of most growth factors are proteins. They can regulate all sorts of cell functions. So we're going to talk a lot about in this course about turning genes on and transcribing genes. And that is essentially completely regulated by a whole series of proteins. They're also responsible for structural formation of the cell, as well as any type of movement that needs to take place either within a cell or the cell movement itself. Antibodies that we see in our immune system are proteins. So it's quite complex, the types of activities that proteins are taking on. And so over the course, we will be talking about all of these different functions that a protein may play. The structure of a protein makes it very unique and makes it capable of doing what it's doing. And so we're going to talk in the next few slides about how a protein takes on the structure that it takes. I'm not going to play this for you right now, but I would suggest you do it. I'll warn you, it's got some really funky music. But it just sort of puts it into context how you start out with these sort of building blocks of amino acids that come together in this big long chain and then fold up on each other in very complex ways to then take on this really distinct shape, which is capable of very unique function. So the building block of proteins are amino acids, and here is the fundamental structure of an amino acid. So at the center you have your alpha carbon. On one side of the alpha carbon you're going to have a carboxyl group. On the other side of the alpha carbon you're going to have an amino group. And this is the same. No matter what amino acid you're talking about, those three things are going to be constant. What dictates differences in amino acids is this R side chain. So you can put a variety of different side chain molecules on here, which will give that amino acid a completely different function, be it interact with other amino acids or other proteins in the cell. Amino acids are sort of going to link, they will link up with one another and are joined by what's called peptide bonds. So in this case, you have the OH, the hydroxyl group of the carboxyl group, combining with the hydrogen of the amino group. So then water is formed and taken off. And now you have the formation of a peptide bond between the C and the N. Typically, a protein or a polypeptide chain is composed of about 450 amino acids coming together and making these bonds. So it's quite a long polypeptide. In some cases, certainly you have a bunch of amino acids linking up, chaining up, but in some cases you also will have other macromolecules combining with a polypeptide chain, giving it even further sort of structural and functional differences. So in some cases you can get carbohydrates, so sugars linking up with amino acids and making what's called a glycoprotein. In some cases you have metals joining with amino acids, which makes a metalloprotein. So hemoglobin, for example, is a good example of that. In some cases, you have organic groups joining up. So in the human system, we are talking about 20 different amino acids that can come together in a variety of different ways to form all the different proteins we need to make. And these different amino acids are different from one another, essentially based on the type of R group that they have. And we can sort of categorize all amino acids into 4 distinct groups. So you can have polar charged, amino acids, polar uncharged amino acids, nonpolar amino acids, and those that have unique properties, which I'll talk about. So the first group are polar charged, and you can see that there are 5 different amino acids here that are listed that fall into that category. So again, if we look at any of these amino acids, they all have the alpha carbon in the middle, a carboxyl group, an amine group, and then in red, it is their R side chain, which makes them very different from one another. In the case of the polar charged group, so this is aspartic acid, glutamic acid, lysine, arginine, histidine, in all cases, what you have are very, very highly charged polar side groups. So even at a very neutral pH, these are usually found in a charged formation. So you can see the O negative here, the NH3 plus here. And this allows them to sort of act, in some cases, as acids and bases, and certainly to form ionic bonds. One that's of considerable interest here is arginine. So histones are those proteins that DNA wraps around. We're going to talk about those quite a bit in our course. Proteins have quite a few arginines, amino acids, included in them. And that's important because you can see the highly positive charge on the arginine side chain here interacting with the negative charge of the phosphate group of the DNA. And that's what allows the DNA to interact with the histone proteins. So I always get the question every year, am I responsible for knowing these different amino acids? And the answer is yes, because as we move forward in the course, I want you to understand when I say DNA is interacting with histones, for example, that the reason that it's interacting is because you have this polar side chain of arginine here that's allowing that to happen. You also have a group of polar uncharged amino acids, so serine, threonine, glutamine, asparagine, and tyrosine. In this case, you can see you still have polar molecules, so you still see your oxygens. Here's another oxygen, an oxygen, a nitrogen, nitrogen, and oxygen. But at a fairly neutral pH, they actually are not charged, so they're very weakly acidic or basic. But they are certainly polar, and they certainly are capable of interacting with other molecules, and especially water, to take part in hydrogen bonding. So these are highly soluble in water. An example that's going to be sort of relevant both to this slide and to the next is over here in the corner. So this is an important protein in our body called cytochrome C, very important in our liver for being able to detoxify a lot of things. And what you can see in this protein, if you look at all of the green molecules that are out here, these are the amino acids that are found on the outside of this protein. All of these amino acids are going to be polar uncharged amino acids. You can see they're found on the outside of this protein, meaning that all the water that's surrounding this protein they're going to interact with that water and allow this protein to become soluble in the cell. Conversely, we have a bunch of non-polar amino acids that do not have polar side chains. And in that exact same protein that we were just looking at, cytochrome C, they're represented here in orange. And you can see they're jam-packed into the middle of the protein, into the hydrophobic area of the protein. They don't need to interact with the water. So you can see there's quite a few types of amino acids that have this sort of a function. So alanine, valine, leucine, isoleucine, thionine, phenylalanine, and tryptophan. And you can see if you look at their side chains, you see no oxygen, you see no nitrogen, so you're not really seeing these polar molecules that are going to set up polarity in the side chains. And as a result, they're very sort of inert, take part. You can see there's a variety of different shapes here as well, which allows them to be really unique and cram into the nooks and crannies in the inside of the protein, where they won't have to have interactions with water. And lastly, there's this group of sort of oddballs that have their own special functions or their own special characteristics. So glycine is special because its side chain, all it is a hydrogen, nothing fancy. But the reason for this is this makes it a very small amino acid, and it allows it to fit into very sort of tight areas. So if you had two polypeptides that need to come into very close contact with one another, you would likely see glycines in those areas because you're not going to have these bulky side chains in the way. Cysteine is very important for protein-protein interactions because it has a sulfur group. If you remember from chemistry, two sulfur groups, when they come in contact with each other, can create what's called a disulfide bridge, which is a very, very strong covalent bond. And so when you have two cysteines that come in close contact with one another, you get this very strong covalent bond that's going to be very hard to break apart. And proline, you can see there is no polarity in this side chain, so it's a hydrophobic in character. But because of its shape, it puts a sort of a kink, and it's a kink in its amino acid. So it actually can add, sort of disrupt the structure of certain proteins or cause sort of a spacing in certain proteins that might be required. So this is what I was just referring to on the previous page in regards to cysteine, which has the sulfhydryl group, which when coming in close proximity with another cysteine can make this disulfide bridge a really strong covalent bond. And it really helps to stabilize some of the intricate shapes of proteins. So let's talk a little bit about the shape of proteins because it's actually quite important. So when we talk about the structure of proteins, there's sort of a hierarchy that we speak of. So the primary structure of a protein is strictly talking about the amino acid sequence that's making up the protein. With leucine beside the valine, beside the isoleucine, etc. So that would be represented right here. So just sort of think about the molecules attached by the peptide bonds. Now, because all of these side groups have different chemistry, it's going to cause the polypeptide chain to start folding in on itself and taking on these really unique structures. Folding that happens is what's called the secondary structure, and it's really the confirmation that those amino acids that are sitting side by side sort of force the polypeptide chain into. The two most common secondary structures that we see are the alpha helix and the beta sheet, which you'll see on the next slide. There's also a few other ones that you may hear over the course of this 2305. So hinges, turns, loops, finger-like extensions, which for the most part are stabilized by hydrogen bonds. So as I said, the two most common sort of secondary structures that we see is the alpha helix. And just as you can sort of imagine, this when you have a very certain sort of alignment of amino acids, in some cases they can sort of wrap themselves up in a helix-type formation. And it actually takes on a very, very structured sort of setup. When you see sort of more simplistic diagrams, which you're going to see in the next couple slides, of an alpha helix, really this looks like sort of a wound ribbon. The other one is the beta sheet, where you have the amino acids sort of folding up and down, and sort of coming around, pulling in 180, and then folding up and down again. And when you see these more simplistically drawn, we see sort of sheets of arrows. And let me just fast forward here, and I'm just going to show them to you over here so you understand. So what you're seeing right here in pink, this would be an alpha helix. And then here where you're seeing these blue arrows, or over here where you're seeing these green arrows, that's representing a beta sheet. So again, you start off with your primary structure, which is just the string of amino acids. Then the secondary structure, the neighboring amino acids cause bits of the protein to start folding into these formations. So you have this alpha sheet, then into a beta sheet, then into another alpha sheet. And then these neighboring sections, which are already somewhat folded, start interacting with each other. And that's called the tertiary structure. So that's when we're starting to look at it in this more complex way. So we can say, oh, well, this protein here has 3 alpha helices and 2 areas of beta sheets. Okay, so that's what we're talking about when we talk about tertiary structure. This type of structure is stabilized for the most part by non-covalent bonds in the R groups. So this can be either hydrogen bonds, it can be ionic bonds, it could even be disulfide bonds, for example. For the most part, we can categorize proteins after they've undergone tertiary structure into sort of fibrous proteins, which take on this sort of elongated format. Typically, our connective tissue is made up of these types of fibrous proteins. So they're a little bit more compact in structure, and those are typically what you see inside of cells. This is an example of myoglobin. Hemoglobin is also very similar. When you look at some of these proteins, as I said, some of them can be 450, even up to 1,000 amino acids in length. So you can get these really huge proteins. And when you get such big proteins, and there's an example here of phospholipase C, different parts of this protein can actually take on sort of specialized functions. So in this case, in the case of phospholipase C, even though this is all one big protein, there are actually 4 sort of subunits built into this protein that each can take on their own unique function. So they actually can all function separately to do different things, even though they're all together one big, huge protein. And so when you have that sort of setup, we refer to these as the domains of the protein. One thing that proteins are capable of doing, particularly enzymatic proteins, is they're capable of changing their shape. So they can have different conformational shapes depending on what has bound to them or what is close to them and changing their chemistry. And this dynamic movement allows them to catalyze enzymatic reactions, for example. Oops. And then finally, we can talk about quaternary structure, so the 4th level of structure. And this is really when more than one sort of protein can come together as subunits. So individually, these little proteins may not be able to function on their own, but when you have multiple of these proteins coming together to make one structural unit, they now have a function. And hemoglobin is really the best example of that, where you have 4 protein chains that come together, 2 alpha chains and 2 beta chains to make the functional hemoglobin protein. If the different protein subunits that are coming together are identical, you have what's called a homodimer, when there's 2, for example, or a heterodimer if they're different. And then slightly different from a quaternary structure is a multi-protein complex. So as I said here, in this case, you have 4 different proteins coming together that are essentially acting as one protein. And individually, they aren't capable of really doing anything on their own. In a multi-protein complex, however, you have a variety of different proteins that on their own are capable of doing some work, but can come together to make these sort of super complexes, which can do lots of different things sort of together. And pyruvate dehydrogenase is that we have 60 polypeptide chains making up 3 distinct enzymes working in close proximity with one another. And I believe your textbook goes over the yeast-2 hybrid assay. This is sort of an old school assay that still is in practice today, but really works on trying to understand how different proteins interact with one another. So the way that a protein is folded really will dictate how and in what way it can function. So there's a variety of things that can happen in a cell which make proteins not function very well. And typically this happens when a protein unfolds. So we call the unfolding a protein denaturation. In a research laboratory, so in my laboratory, when I'm trying to measure proteins, some proteins can compact themselves so tightly and so small that it's really hard for us to actually measure them. So what we would do in the lab setting is we would denature them somewhat, so we would start to unfold them a little bit, just so that we can actually easily measure them. So we can do that in the lab by using detergents, or reducing agents, for example. And if anyone has ever done a Western blot, you typically are going to be using these things to try and open up the proteins a little bit. Important to think about in regards to environmental exposures. So radiation and heat can also denature proteins. So when you think about different environmental exposures that could be damaging some of the proteins in cells, you can start thinking about disease processes there. The other aspect related to disease and protein folding, probably something that you guys may have heard of in the past couple of years, because there's been a few cases here in Canada that have cropped up in the past several years. And this is diseases that are entirely driven by a prion, which is basically a protein causing disease. And this protein that's causing disease is a protein that is actually, from an amino acid sequence, is totally normal. It absolutely should be in our bodies. But in the case of Creutzfeldt-Jakob disease, this protein, the amino acid sequence that is totally normal, comes together and does not fold properly. And because it cannot fold properly, it actually starts clumping up in cells and causes cells to start dying. And typically what we see in this case is that this misfolded protein is happening in the nervous system, primarily in the brain, and you start to get lesions forming in the brain. So a lot of the neurons start dying, you get lesions. And unfortunately, this is a very progressive and fatal disease, so we have no way of fixing this once this starts. So as I said, this is a protein. It's called the PRP protein, which is typically found in healthy brains. But the form of the protein that we see in healthy brains is PRP, denoted with a C, and it's this protein right here. So you can see we have 1, 2, 3 alpha helices and 1 beta sheet. That's sort of the folding structure that we see in this protein. However, for whatever reason, these individuals who are getting this disease, the same amino acid sequence ends up folding in such a way as to have 1, 2 alpha helices and instead quite a few beta sheets. This protein is no longer soluble in the cell. It starts to clump up and glutinate and starts gunking up the cell essentially and causing the neurons in the brain to die. Very similar diseases that seem to be working along almost the same line as mad cow disease, Guru and Scrappy. The last series of proteins we're going to talk about is on this slide right here, and those are molecular chaperones. And these are really helper proteins. So these proteins their whole job is to try and protect other proteins that are found in the cell. Protect them from damage, from stress that might be happening, or just to protect them as they undergo that undergo that folding. Make sure that they do fold properly. So for example, the heat shock 7B protein, so that's these chaperone, these little purple proteins here, they have a really interesting job. So as you have your ribosomes, which are driving along the mRNA and making these big long polypeptide chains, which are not yet folded, they will essentially come and grab onto this mRNA here, and they will force this mRNA to fold properly and not be disrupted by any of the other things that might be going on in this cell. So you get really proper folding. There's also another type of chaperone proteins, chaperonins or TRIC-C, and this actually almost acts like a little protective chamber. So this mRNA, after it gets split out from the ribosome, can enter into the hollow center of this protein, and it can allow all the folding of the protein to take place inside so that it, again, does not get disrupted by anything else that's going on in the cell. It's pretty amazing little proteins that are making sure that this job is done properly. And then the last thing, which we're not going to chat about too much in this lecture, your book goes into great detail, and for those of you that are using an older edition of the textbook, I would recommend looking at the newer editions for this section here on proteomics. So this is really a field of molecular biology where you can investigate this and study the entire proteome of an organism. So instead of going in and looking at just one or two proteins, you can actually assess all the different proteins that are in a certain cell, in a certain tissue, in a certain organ, for example. And you can compare, if you wanted to, a healthy organ to a diseased organ. There's a variety of different methodologies that allow us to do proteomics. So gel electrophoresis or microarrays or mass spec are some of the more common ones. But the textbook really goes into a lot of detail on the methodology. We do a little bit of proteomics in my lab, and when we give you the lecture on the research going on in our lab, we will touch a little bit on some of the proteomic methodology. So that is it for today's lecture. So on Wednesday, Dr. Keir Menzies will be coming in to give you guys the lecture on metabolic pathways. He is the prof of the other section, so you'll be getting the same lecture as the other section is getting. And I will be back on next Monday to give you the fourth lecture. So with that, I will wish you a good week, and I will see you next week.